Great. Oke. Terima kasih kepada Bapak Ibu yang telah bersedia untuk hadir dalam acara ini. Sorry, Petros, I will say it in Indonesian language a little bit.
That's fine.
Okay, great. Terima kasih Bapak Ibu yang telah bersedia hadir dalam acara ini. Sebentar lagi kita akan memulai webinar dengan pembicara oleh Profesor Petros Ligoxygakis dari University of Oxford, dan materinya berhubungan dengan Trypanosomatid disease menggunakan animal model Drosophila melanogaster, lalat buah. Beliau akan membawakan materi dan saya akan men-share sedikit flyernya sebelum saya persilakan kepada Ibu Yulia Yusrini Jabir untuk memberikan ini pengantar. Jadi Profesor Petros Ligoxygakis ini akan membawakan materi Trypanosomatid Parasites and Insect Immunity. Dan insya Allah dalam beberapa menit kemudian kita akan mendapatkan pandangan yang mungkin tidak lazim ya di Indonesia menggunakan animal model atau model organisme lalat buah Drosophila melanogaster. Oke. Thank you for your patience, Professor Ligoxygakis. Now I will proceed to the opening remarks by Ms. Yulia Jabir, PhD. Ms. Yulia Jabir, are you okay? On?
Yep.
Okay. Yes.
I can hear you clearly.
Okay. Now we just this is casual, I think. Just okay. Please, the floor is yours. Thank you.
Okay. Thank you very much, Mr. Frisanaino. And what a pleasure today that I'm giving these opening remarks. But actually, our dean would like to send his apologies to Professor Petros Ligoxygakis because he couldn't give these opening remarks today. But I think he will join a little bit late. And firstly, of course, on behalf of our faculty, we would like to send our gratitude to Professor Petros Ligoxygakis from Oxford University because he will share about his knowledge and research topic which pretty new to us, and probably we could use it for research collaboration next time. I don't know, but we'll see what's gonna happen next. And I would like to tell everyone that this is our third webinar series that is moderated by Pak Frisanaino, Mr. Frisanaino. So I'm really glad that Mr. Firsan can also collaborate with lots of international researchers, and that he will help more webinar series in the coming future. So I would like also to thank all the participants that has coming today. And so without further ado, I would like to give back to moderator. Thank you.
Thank you very much, Ms. Yulia Jabir, PhD, for the opening remark. And yes, without further ado, I will also introduce Professor Petros Ligoxygakis to everyone. Please let me share the PowerPoint slide to make everyone aware of the event. Okay, great. So yes, today the speaker, as I previously mentioned, is Professor Petros Ligoxygakis from Department of Biochemistry, the University of Oxford, and I will serve as the moderator in this session. In this webinar, Professor Petros Ligoxygakis will discuss recent findings from studying the interaction between Drosophila melanogaster and its natural Trypanosomatid parasite, Herpetomonas muscarum, and their relevance to studying a more medically important interaction, that of Phlebotomus papatasi, the sandfly, and Leishmania major. For a brief introduction about Professor Petros Ligoxygakis, in 1995 to 1999, he did his PhD with Petros Ligoxygakis and Fotis Kavatos in Drosophila development at the Institute of Molecular Biology and Biotechnology, Crete, Greece. And 1 year later, he did his postdoc for 4 years with Jules Hoffman, which received Nobel laureate in 2011 in the field of Drosophila immunity. So I think his credential is really amazing with this. And then 1 year later, he got admitted as a lecturer of genetics in the Department of Biochemistry, the University of Oxford. And since 2013, he served as the professor of immunology in the Department of Biochemistry and fellow at Hertford College, both at the University of Oxford. He served as the fellow of the Royal Entomological Society. So I believe with this amazing experience, he can share us some basic understanding about Drosophila and also pathogen. Okay, Professor Ligoxygakis, I would like to give the speak to you. Yes, you may speak for now. Thank you very much.
Okay, let me share my screen. Can you all see that?
Yes.
Perfectly.
Yes, we can.
Okay, can you see my pointer?
Yes.
Yes. Fantastic. Okay. First, thank you for the kind introduction and for your invitation, and most of all, thank you for giving me the chance to communicate our work with the students and researchers in Indonesia. So we are a insect immunity lab, and why insects? Well, we are very interested in insect immunity because, from pollinators to vectors that transmit deadly infections, the infectious diseases, or support life in the rainforest, insects are extremely important. And their immune system is one of the major ways they have to interact with the world around them. So understanding this will help us understand in turn how life on our planet in many different contexts and in various ecosystems is supported by insects. So for the purpose of this talk, we're going to discuss how insects interact with Trypanosomatid parasites. And Trypanosomatid parasites are protozoan parasites and include some of the highest impact human and animal pathogens. However, the treatment and prevention options are very limited. And here you see some of the diseases that Trypanosomatid parasites cause. Now, of course, Trypanosomatid parasites are, very few Trypanosomatid parasites can be passed on from insects to humans, right? Most of them just infect insects, and there is a evolutionary relationship between its parasite and the host, and this is the side of the host evolutionary tree, and this is the side of the parasite, and you see that the whole the evolutionary relationship spans almost 300 million years of evolution. So we call Trypanosomatid parasites that infect only insects, we call them monogenous parasites, where parasites or Trypanosomatid parasites that can infect both insects and mammals, we call them digenous parasites. And the two genera that are responsible for the vast majority of the burden in both human and agriculturally vital livestock species are Trypanosoma and Leishmania. Right, so Leishmania infects Phlebotomus and Lithomia genera, which are sandflies, and Trypanosoma infects Glossina, which are the checheflies. And Trypanosoma causes the human and animal African trypanosomiasis and Chagas disease, or American trypanosomiasis, as it's called, and Leishmania causes leishmaniasis. But keep in mind this evolutionary relationship, both from the side of the insect and the side of the parasite, because it will be very helpful when we're going to discuss the relationship between Drosophila and its natural Trypanosomatid parasite Herpetomonas, and how we can use it to study this relationship between Phlebotomus and Leishmania from both sides of the host-parasite interaction. So just to give you some numbers on the burden, what I call the burden of the disease, and these are numbers that were included in the Global Burden of Disease 2017 report that was commissioned by WHO, and you know, estimates of incidence and mortality vary. But some studies predict that for leishmaniasis, which is going to be the focus of my talk today, more than 4 million people had the disease in 2017, including almost 700,000 visceral cases. So visceral leishmaniasis is the most serious aspect of the disease that can prove almost 95% fatal. And also the calculation includes 1 billion people at risk of infection. Now, but what I want to emphasize here is this metric that the WHO introduced this decade that are it's called disability adjusted years, and the disability adjusted years is a metric that gives the burden of those people that haven't passed but are chronically infected. And then you will see this as an additional information about the vicious circle of disability, poverty, and disease that these infections occur. Now, to reduce infection and break the vicious circle of poverty and disease in affected communities, this requires various public health, social, and financial interventions and aid programs. And these have been effective in reducing pathogen loads. However, to truly eradicate diseases such as leishmaniasis, we should target the insect vector before it passes the pathogen on to humans, thus cutting the lines of transmission. And I believe that to achieve this, we need to understand the basic biology of how the insect immune system interacts with the microbial world. And most of our knowledge for this interaction of the insect immune system with the microbial world is from Drosophila melanogaster, the fruit fly, which is a model system with unparalleled genetic, genomic, and bioinformatic, as well as in vivo study tools, underscored by more than a century of fundamental research. And most Drosophila immunity studies have focused on bacterial, fungal, or viral infections, but lack data on interactions with trypanosomatid parasites, which through other more medically important but related species cause some of the highest impact insect-borne diseases, as we said earlier. So we wanted to see whether Drosophila had trypanosomatid parasites as a pathogen or symbiotes in the wild. And this is a study on the distribution of various microorganisms in wild populations of Drosophila melanogaster in Europe, both southern and northern Europe, and you can see that Leishmanian trypanosoma, which are not actually Leishmanian trypanosoma, but are trypanosomatid parasites that just look very much like Leishmanian trypanosoma, and in fact are Herpetomonas, and you will see in a minute why I think that. You can see that many different populations of Drosophila have this in the wild, and this is a paper that's going to come out very soon. This is from a consortium, a population genetics consortium that has studied this. So these are therefore parasites that can be found naturally in wild populations in Drosophila, and therefore we can try and study them to see whether we can gain insights into other insect trypanosomatid interaction. So one of these parasites that infects Drosophila is called Herpetomonas muscarum, and it's a trypanosomatid parasite, and you can see if I don't know why I'm not able to play the movie. Let me see. Let me play the movie like that. So you see, so this is the walls of the guts. You know, this is the visceral muscle here, and this is the epithelium, and this is the gut lumen. And if you look, this is a kind of low resolution video that shows the parasite kind of attaching into the walls of the guts. Now, it's nevertheless cleared by the fly, and you will see here that 54 hours after uptake, you see the very small amount of parasites that are left, and after 100 hours they're all cleared. And the effect that the parasite has on the fly is quite striking. So the flies lay less eggs. So this is the laying of eggs of a wild type fly, and this is the laying of eggs of a wild type fly that is infected. And they lay even less eggs if they are immunocompromised. Right, so this is in the green line here, the laying of eggs of an immunocompromised fly. So these are significant different numbers between infected and non-infected flies. But what happens here is that this reduction in investment in egg laying means that the fly, an infected fly that clears the infection, is able to live longer. So this is the lifespan of a fly that was fed just for 6 hours with sucrose, and this is the lifespan that has been infected for 6 hours with sucrose and a dilution of Herpetomonas muscarum, which is, quite staggering that an infection will have this kind of effect. However, less egg laying means that the infected fly has less chances of passing on progeny. Now, the other important thing to observe is that the bacterium, the microbial flora, is not really influencing the lifespan of the infected flies. So the power of the Drosophila model is that we can raise germ-free flies relatively easily. So germ-free flies means flies that are in a sterile environment, eat sterile food, and they are themselves sterile because we have bleached the eggs and put them in sterile food right from the beginning, right from the start of embryogenesis. And therefore, these are true germ-free flies. And you see that infection doesn't really influence the germ-free lifespan. Of course, germ-free flies, it's well known that germ-free flies live less than normal flies and conventional urea flies, but this difference is not influenced by Herpetomonas infection. So we have developed tools to image and genetically manipulate the parasite. As you see in this slide, we were able to produce stable transfected lines for both imaging and deletion or no-king mutants. So this is a stable transfected line, and you can see that the stable transfected lines and the wild type parasite has the same almost identical behavior and numbers in the fly. And with this, we can track how the parasite actually travels within the fly gut. And let me just introduce to you the fly gut. So this is a schematic of our gut, of the mammalian gut, where from the esophagus you go to the stomach, large intestine, small intestine, and so on. So the crop is this here, and is the insect equivalent of the stomach. And there is the midgut, which is the insect equivalent of the small intestine. And there's the hindgut, which is the insect equivalent of the large intestine. And upon uptake, you see, so these red ones here are the parasites that are gathered in the stomach, and then gradually they make their way through the tubing of the stomach to the midgut, and then to the posterior midgut, which is a blow up of this region you see here, and you see the red red dots, which are the parasites. Now, this is again a continuation of the journey of the parasite, and again you can see the contractions of the gut and the parasites inside the gut, and here you see parasites attaching themselves or trying to attach themselves into the midgut epithelium. right? And this is a blow up of this area of the posterior midgut through colonization. Now, we also see that Herpetomonas bring about formations that we call rosettes, and they are like that. So you see the parasites here that gather together. This is a zoom from the lumen of the gut. And you see that the parasites are gathered together in a formation that is reminiscent of Leishmania, Leishmania parasites. Now, we sequenced the genome of Herpetomonas, and this is kind of the typical Trypanosomatid genomes, and you know, you see the data there. It doesn't say much, but just to say that it's kind of the expected size of the of a Trypanosomatid genome. And the relationship between Herpetomonas muscarum and other Trypanosomatids, the evolutionary relationship, of course, brings Herpetomonas closer to Leptomonas and Crithidia, and then immediately closer to Leishmania. However, we found that this difference is very small, as you see here, that they have slightly less common genes between Herpetomonas and Leishmania, and Herpetomonas and Leptomonas. However, in terms of synteny, which is genes remaining at the same position on corresponding chromosomes and in their corresponding order, which is collinearity, we find, so synteny is the position of the genes in the chromosome, and collinearity is the corresponding order of the gene in the chromosome. We found that there is a remarkable collinearity and synteny with Leishmania, despite the fact that they're supposedly, Herpetomonas is supposedly closer to Leptomonas and Crithidia. And you see here, so this is the classic sort of basketball image of synteny and collinearity. So this is a Leishmania chromosome, and this is a Herpetomonas chromosome. And wherever there is synteny and collinearity, you will see a line from the gene from Herpetomonas to the gene in Leishmania. And just by looking at this, you see how synthetic this chromosome of Leishmania major is to Herpetomonas muscarum, and this is just you know, this is just one example. You know, all the chromosomes have this kind of very extensive synteny and collinearity, which actually we can quantify, and you see that Leishmania major is very much and significantly more synthetic than any other of the other Trypanosomatids. Now, another thing that we did is that we elucidated the gene expression of the parasite in vitro, but also inside the fly. And that's very important because, as you will see, there is a marked difference between what's happening in vitro in terms of the gene expression with what's happening in vivo. For example, if you compare this side of the graph with this side of the graph, or this side of the graph with that side of the graph, you will see that the dynamics of gene expression during infection is very different than the dynamics of gene expression within a culture. And this you can see also in this PCA plot, where the in vitro gene expression is totally different and grouped very far away from the log phase or stationary phase parasite. Nevertheless, what was again very revealing was that there were real parallels of gene expression within the fly with Leishmania major, making Drosophila and Herpetomonas a good model to understand what's happening in the Sandfly Leishmania interaction. Now, I talked about in fly transcriptomics, and of course in fly transcriptomics happens inside the animal, but the infection, the root of infection, is very important. And let me just go through a little bit of how this happens. So this is a schematic of the fly gut, and you can see the enterocytes. So these are the enterocytes with the villi, the microvilli of enterocytes, which are the workhorses of the gut, as well as the enteroendocrine cells, which coordinate the gut response to systemic changes, metabolic changes, changes of stimuli, and facilitate the interaction of the gut with the brain. And finally, in the basal membrane, very close to the visceral muscle, are the intestinal stem cells. So the intestinal stem cells replenish enterocytes, so they divide to give themselves, and then an enteroblast that will eventually either give an enterocyte or an enteroendocrine cell. And when there is an infection, but also in terms of the normal flora, duox is a very important, dual oxidase is a very important enzyme that through calcium coming from the ER will help produce reactive oxygen species, ROS, to counter bacteria, and then also enterocytes through the IMD, NF-kappa B, RELIS pathway will produce antimicrobial peptides, AMPs, and also peptidoglycan recognition proteins that have amidase activity and that will degrade the immunogenic potential of the bacterial cell wall to keep the homeostatic conditions in the gut. So in this context is where infection with a Trypanosomatid happens in any insect, and especially in our example here, Drosophila. And what happens, I mean, this is a very busy slide, and it summarizes years of work. But what I want to emphasize is that here, I think I'm doing something with my. Okay, so and emphasize the dynamics of infection, and you see here the time points that gene expression was taking, genes that were upregulated, genes that are down-regulated, and there are different genes that are up and down-regulated, as you see here in different time points. There are many genes that belong to the immune category, like antimicrobial peptides and other immunity-related genes. There are many genes that have to do with digestion, proteolysis, lipid metabolism, carbohydrate metabolism, and so on and so forth, but also neuropeptides to coordinate the response in the whole organism. So what we did is that we tried to see which of these genes are important for the work of the enterocytes and the intestinal stem cells, to see how the infection was influenced in the gut. And with Drosophila, the power of the system is that you can do RNAi to specifically knock down genes in either enterocytes or intestinal stem cells. And here, what we have done is we have knocked down DUOX. You remember that is important for reactive oxygen species in enterocytes, and you see that this generates an increased uptake and slows down clearance. And this RNAi is effective at 30 degrees, but not effective at 18 degrees. And these are the two temperatures that we are comparing. And of course, to emphasize that we have done the crucial control to compare the infection of wild-type flies or non-RNAi flies between 30 and 18 degrees, and nothing happens. So there's no difference. So the difference that we see has to do with the RNAi and not with the just the change of the temperature. And also we can inactivate antimicrobial peptides. Dipterosin and sacropin are two antimicrobial peptides that we can inactivate and see the increase in uptake. And there's a trend in slower clearance. And conversely, if we do the reverse and overexpress antimicrobial peptides, we see that there is a decreased uptake and a very fast clearance of the parasite. Now, STAT is a transcription factor, which is important for intestinal stem cells, and we see that if we knock it down in enterocytes, nothing really happens. But if we knock it down in intestinal stem cells through, actually intestinal progenitors through the escargot line, we were able to increase the uptake and slower the clearance. So STAT as a transcription factor must play a role in intestinal stem cells. And indeed, upon infection, a marker that has 10 binding sites of the STAT transcription factor linked to the stabilized GFP is increased markedly following infection, and is at a peak at day 3 of infection in a comparison to a sucrose feed control. And this correlates with an increase in the in the division of intestinal stem cells, and the division of intestinal stem cells we can find out by an antibody against the phosphorylated form of histone 3. So dividing cells will have a lot of pH3, and you see that upon infection there is a marked increase that comes at a peak at 30 hours of pH3 plus cells. And this also correlates with the increase in intestinal progenitor cells, and this is a marker that marks intestinal stem cells and enteroblasts, and you see that again there is a correlation between intestinal stem cell progenitor numbers, intestinal stem cell division, and STAT activity. So what we also wanted to do and have done is to look at, I said that in, let me go back, and I said here that there is Duox and JAK-STAT. So Duox that is regulating reactive oxygen species, and JAK-STAT that is regulating intestinal stem cell proliferation, but there is also the IMD pathway that is regulating antimicrobial peptides, amidases, and other responses. And we wanted to see the role of RELIS and IMD in that kind of infection. And again, you see the surge of intestinal stem cell proliferation measured by an antibody against phosphorylated histone 3, and this is suppressed in a RELIS background, and also suppressed in an IMD background 6 hours following infection, and this suppression is sustained 1 day after infection, and RELIS is the Drosophila homologue of one of the Drosophila homologues of NF-kappa B, and it is indeed still the case 3 days later that there is a marked suppression of intestinal stem cell proliferation when actually, in the wild type situation, intestinal stem cell proliferation has come at its peak. And the fact that RELIS NF-kappa B is important for intestinal stem cell proliferation shows here in this slide that if we overexpress RELIS, whatever the condition of the fly, either sucrose fed or infected, we force cell division of the intestinal stem cell. So our model here is that the STAT transcription factor and the NF-kappa B transcription factor are all important for intestinal stem cell proliferation and enteroblast generation of enteroblasts, and then antimicrobial peptides and Duox are important for intestinal and enterocyte function. Now, as I said, what we really want to do here is to be able to use the knowledge that we have gained from our studies in the interaction between Drosophila and Herpetomonas, and study this insect. And this is a schematic of a Sandfly infection, Leishmania major infection in sandflies. And what happened is that the insect will bite you, And the blood meal will go to the midgut lumen and will start being digested. And the parasites, as you saw also in the videos with Hepatomonas, will try to attach to the microvilli of the gut. And then this attachment will establish the infection. And what will happen is that after the digestion of the blood meal, the parasites will travel to the salivary glands and the mouth parts of the insect, waiting for the next bite that will be transmitted then to the next individual. And what we did with our collaborators in the Parasitology Institute in Prague is that we have studied sandflies that have come from this area of the mountainous area of Morocco. This is the average elevation of the Azila province, and Taza is also a mountainous area where visceral leishmania and cutaneous leishmania coexist, and has been the focus of many interventions to try to reduce the lines of infection since a decade ago, let's say. So what we found is that first what we did, so we gave these sandflies only blood, which was our control, Herpetomonas muscarum, or Leishmania donovani and Leishmania major. From all of these parasites, only Leishmania major is able to establish infection in those flies. But what you can see in this principal component analysis of gene expression following a blood meal, post-blood meal, 1 day, 4 days, or 9 days, is that infection doesn't really play a role. What plays a role is the blood meal and the time, right? And you see the gene expression is clearly separated between 1 day post-blood meal, 4 days post-blood meal, and 9 days post-blood meal, right? And that was a surprise to us, and it's also reflected in, if you try to heat maps to be able to look at immunity genes or signaling immunity genes or antimicrobial peptides or other regulators of immunity. And you see here the different treatments, 1 day post-blood meal, 4 days and 9 days post-blood meal. And you will see that, through all these treatments, there is quite a similarity, and any difference is not really statistical significant. Look at this, for example, 1 day post-blood meal, there is a huge similarity in how, regardless of infection or not, is the gene expression similar. And the same thing goes for oxidative stress and STAT signaling, which is very important for Drosophila, as we saw, and for Duox. So all of these genes that we saw were upregulated in Drosophila are also upregulated in sandflies, but not because of infection, but because of blood feeding. And in a way, although we were surprised at the time, this shouldn't be surprising, because the blood that a blood feeding insect can accumulate, and especially sandflies, can be 3 times its weight right, in one blood meal. And of course, this is the most important physiological change the sandfly reacts to, rather than the presence of a parasite, because the concentration of the parasite within the blood meal might not be so that would activate over and above the immune response that is activated following the blood meal. So the parasite, in the end, might be just one in the one feature in the microbial landscape of the insect gut. And I think, again, I want to leave you with this sort of conclusion, that this result will generate new thinking around the concept of stopping transmission by controlling the parasite inside the insect, because maybe what we need to control is the blood meal itself, rather than the parasite inside the insect. And with this, I would like to acknowledge the people in the lab. Megan has done a wonderful job in elucidating the gene expression in sandflies and Herpetomonas, and did all the genetics of the Herpetomonas parasite. And Li, who is unfortunately not in this picture, is a postdoc that left the lab, did all the Drosophila work and gene expression in Drosophila, and has isolated the parasite of Herpetomonas in the wild. And we have been collaborating with Peter Wolf in Charles University in Prague, and with James Cotton in the Wellcome Trust Institute in Hinxton in Cambridge, here in the UK. And these are our funders. So thank you very much for your attention, and I'm ready to take questions. Thank you.
Thank you very much, Petros, Professor Ligoxygakis. Actually, this is really new for most of us, because Drosophila is pretty new model organisms in Indonesia, except in biology department. So I believe everyone have new understanding about this interaction, host pathogen interaction, using Drosophila model system. Okay, great. I just want to announce something to everyone in here in Indonesian language, if you don't mind.
Yeah, of course.
Okay, thank you very much. Okay, Bapak Ibu semuanya, jadi presentasi telah diberikan oleh Profesor Petros Ligoxygakis, dan sekarang adalah sesi untuk berdiskusi. Bagi Bapak Ibu yang ingin bertanya, dapat langsung bertanya ke Profesor Petros atau melalui saya sebagai moderator. Bisa menggunakan bahasa Inggris, atau menggunakan bahasa Indonesia, nanti ditranslate. Oke, panitia menyediakan, sekedar informasi bahwa panitia menyediakan 2 hadiah untuk 2 penanya terbaik yang akan dipilih oleh Profesor Petros Ligoxygakis. Terima kasih. Oke, everyone. Please, if you have questions, please raise your hand or just ask directly to Professor Petros. Is there anyone who wants to ask questions? Okay, as a start, maybe I will ask questions.
Okay.
Okay, great. So while waiting, because maybe they ask in chat, for committee, please kindly let me know if someone asks in the chat, but I may not see it. Okay, Petros, actually, I'm wondering about your presentation. You showed that IMD pathway and duox and stats is kind of like playing a role in the outcome of infection, especially in the intestinal stem cells. But when I saw your data, it shows that in the early time of infections, toll pathway is activated, isn't it?
Yeah.
Yeah. Do you know why toll pathway is activated and then later changed into MD pathway?
So we have done both gene expression analysis in guts and in whole flies, and we find that it's the whole flies where toll pathway is activated, and that I mean, both in the fat body, which is the insect equivalent of the mammalian liver, and in the intestine, toll is needed. So in the intestine is needed in the enterocytes, and in the fat body is needed for antimicrobial peptide activation. So for the gut, and I mean, I didn't want to go into details because that's a whole different story. We find that toll is playing a role in controlling the metabolism of the intestine through a translational regulator that's called 4-EBP. And this is the role of toll. I mean, I don't want to say anymore because this is something that we are working on at the moment, and maybe there will be things that I will say that it won't be true, right? And
Okay.
we will find that it won't be true. But we have found. Let's put it this way. You're right that toll is activated, and the data show that toll is activated, and toll is indeed playing a role not just following parasite infection, but following other kinds of infection, and also in the homeostatic conditions. And we're trying to figure out how this might be. But our now working hypothesis is that toll is regulating metabolism in the fly, as well as, of course, antimicrobial peptides and the classical immune defense that we all know.
Great, thank you, Professor Ligoxygakis. Maybe someone wants to ask questions. I just don't want to get the present by myself, because if only me asking question, then I will get the present, isn't it?
Hello, Mr. Fursan.
Okay. Please go ahead, Mr. Dian Permana.
Yeah, okay. Thank you very much, Professor Petros, for a really nice presentation. It was really good. So basically, my question maybe will not be related to the topic that you have explained, because it's kind of pretty new for me, because I'm working on pharmaceutical technology and drug delivery system. But once you explained about all those things, one thing just came to my mind, like, do you have any experience working on research collaboration with someone or research institute who is focusing on formulation or drug delivery, and then they applied their formulation or their system into the model that you have developed in your lab, for example? So can you just share this kind of experience to us? Maybe it's just.
I mean, yeah. Well, thank you for that question. This is a very important question. I think our goal. I mean, we're not there yet, but our goal is to be able to use. If we know enough about the basic biology, about the interaction of the insect with the parasite, the goal is to use compounds in the food in the beginning, or in the blood, or to be able to suppress the immune response of the insect, or increase, in any case, manipulating the immune response of the insect, or prevent the parasite from binding onto the walls of the gut, so it cannot establish infection. Because in essence, what is important here is that the bottleneck, the restriction for infection and transmission is the parasite establishing infection inside the insect. If this doesn't happen, there is no transmission, right? The insect lives, but the parasite doesn't. And we want, I mean, we're not there yet, as I said, but we want to test compounds that will be able to either help the insects withstand the infection, or prevent the insect from digesting the blood meal, or you know, trying to manipulate the immune response of the insect. But we need to know more about the basic biology, right? So this is why we're doing this. But you're right that in the end, what we want to do is to find ways of and compounds to manipulate the parasite presence or the blood presence inside the insect.
All right, thank you very much.
Okay, thank you, Mr. Dian Permana. So Mr. Dian actually graduated from UK, isn't it, for PhD?
Yeah.
Yeah. Okay, is there anyone else who wants to ask questions?
I would like to ask a question.
Okay, please go ahead, Miss Dini.
Okay, thank you very much. So maybe I want to ask more towards the general question, like very basic information, because the when Drosophila infected by parasite, till what extent that we already know what kind of pattern that Drosophila, I mean, like the immune reaction of Drosophila when it's infected by some parasite. In the previous work, I worked with virus, when virus infected Drosophila, but I think this is a kind of new pattern. When parasite infected Drosophila, it's kind of like have a different pattern comparing to the when it's infected by virus. Could you please explain the basic knowledge how the Drosophila react when it's infected by parasites? Thank you.
Yes. Well, this is the model that we have, right? That when there is so it's very important to understand the root of infection, right? The way the insect is infected is through the gut, right? And this infection will activate intestinal stem cells. And this intestinal stem cell activation will require the STAT and NF-kappa B transcription factors that will then influence enteroblasts and enterocytes. And TOL and DUOX and antimicrobial peptides are important for enterocyte function, right? So STAT and RELIS are important for intestinal stem cell function, and TOL, DUOX, and antimicrobial peptides are important for enterocyte function upon infection. But what you see here, that what's happening following an infection is that, so this is the parasite infection, this is a sucrose feeding. You see that there is a surge in intestinal stem cell proliferation, right? And without relish, then this intestinal stem cell proliferation doesn't happen, right? And also without STAT, this intestinal stem cell proliferation doesn't happen. And you see here how STAT is important for the activation of STAT dependent transcription. So this is our basic model, right? That there is an infection through the intestine, through the gut. NF-kappa B and STAT activate transcription in intestinal stem cells, and then you need duox. And let me go to, so you need duox and antimicrobial peptides in the enterocytes to counter the infection, right? So STAT and NF-kappa B in intestinal stem cells, duox and antimicrobial peptides in enterocytes.
So, how's Dini?
Okay, thank you.
Maybe Professor Ligoxygakis' connection is disrupted? Is it? Okay.
Can you hear me?
Yes, we can hear. So what about the answer, Dini? Is it okay for you? I mean,
referring to the diagram that it states after 12 activated AMP pathway, and then what happened after the AMP pathway activated? Actually.
Well, yeah, that's a good question. So what we think happens is that the turnover of intestinal stem cells, the turnover of enterocytes and the proliferation of intestinal stem cells will start killing the parasite and will prevent the parasite from attaching long-term to the gut. I can maybe go to the
sorry, Rizka, please mute your. Okay, sorry, Petros.
Yeah, it's okay. So let me just play this. So you see that the parasite is trying to attach. Can you see that? Yeah. And we think that the turnover of the enterocytes and antimicrobial peptides and reactive oxygen species prevent this attachment. And also, if you see here, the gut is contracting. Can you see that?
Yeah,
that the gut is contracting. So these contractions will push, the parasites eventually, the parasites that cannot attach will push them out, basically. And this is how clearance happens.
So the mechanism makes the parasite cannot penetrate outside the gut, isn't it?
Yes, cannot attach to the gut, because we don't think that the also leishmania cannot really penetrate, just travels through the gut epithelium to the mouth parts of the insect and then is transmitted.
Okay, so there is no rules of hemocyte involved in this mechanism.
That's a good question again. We don't know. We have done a few experiments to knock down things in hemocytes and see what happens, and we haven't seen any effect. So as far as we know, and for the genes that we have tested, there is no role. But I mean, I cannot say for sure that there is no role 100%, right? Because there are things that we haven't tested yet.
Okay. Thank you very much for your answer.
Thank you, Dini. So Petros, actually, Dini and I worked together when we were in Kanazawa, and that's why she asked probably hemocytes, because we work with hemocytes role in the virus infection. Thank you, Dini.
Yeah, thank you very much for your answer.
Great. Another questions, perhaps?
Maybe from me?
Okay. Please, Ms. Yulia.
Okay, thank you for the opportunity. I'd like to ask about the reaction, so the signaling pathway following the parasite infection in Drosophila. Do you think that also apply in humans? in that way, I mean, it's a really good opportunity for us to see what happens and what, you know, the therapeutical target for probably. What do you think?
What do I think? I mean, these infections are different in humans and in insects in the sense that the parasites are in different environments. So the parasite in insect is in the gut. The parasite well, Herpetomonas doesn't really infect humans anyway. Okay. But Leishmania, for example, is not, you know, is not in the gut. Is in blood cells, and you know, and is countered by adaptive immunity, and also has a different developmental stage. So I don't think we would be able to directly compare what's happening in the insect and what's happening in the in humans, because there are, you know, the parasite is in a different, Leishmania is in a different developmental stage, and also the environment, the tissue environment is, and the cell environment is completely different. But what you could say is that there is a very interesting host pathogen interaction problem here. You know, a three-way interaction. There is a mammalian host, there is an insect host that can accommodate the same parasite in different life stages, right? And the, you know, evolutionary changes that have to be done in the parasite to be able to adapt to both of these hosts are really very impressive, very remarkable, and I think make the fascination of people for this kind of, you know, interactions, right? Because there's you know, a great deal of very exciting biology that can be discovered.
Okay, thank you.
Thank you, Ms. Yulia. Maybe it's been nice to hearing some interesting questions, isn't it?
Yeah, absolutely.
Yeah. Is there any questions? Oh, maybe not so many. Okay. Mr. Akbar?
Hello, Petros.
Hello.
I'm interested actually in microbiomics, about microbiota. And I don't know whether in fly there is the microbiota in the gut of the fly will affect, whether it will affect the interaction between the host and the parasites. What do you think?
So as I said, we don't see really an effect. So flies are good because you can grow germ-free flies. So flies that have sterile embryos, that you put them in sterile food and in a sterile environment, and you can have what we call germ-free flies, right? And you can see that the infected germ-free flies and non-infected germ-free flies, you know, don't really have a difference in lifespan, and maybe in the, you know, they have a difference from conventionally reared flies, right? Germ-free flies in terms of their lifespan, but this is not due to the infection. Right, this is just due to the fact that they don't have microbiota, and microbiota we all know is good, you know, in a healthy individual is a good thing. So I don't think that microbiota influence the defense of the fly in this particular conditions, but we know that microbiota are important. So the bacterium is important for Leishmania infection in sunflies. And we know that because with the blood meal, imagine, you have an influx of a huge amount of blood inside the insect gut, the microbiome changes because there is a lot of nutrients that are coming in and bacteria start proliferating. So the interaction between this phenomenon and the attempt of the parasite to establish an infection is a very interesting one. And for sure, it has been shown actually by Rod Dillon's work in Lancaster here in the UK that the microbiome is important.
Thank you.
Okay. So, Petros, based on this figure, I remember that you said that in the presence of infection, the number of eggs laying by the female is decreasing, right? Decrease. And then at the same time, they also have a longer lifespan.
Yeah.
Is it? Okay.
Yeah.
Is it common in the like parasite infection?
I mean, it's not common in parasite infection, but you can, explain it because there's always a trade-off between lifespan and fertility and investment in, in progeny. So if there's not so much investment in egg laying, then, you could expect that the flies that don't lay so many eggs will live longer. Of course, the caveat here is that if you don't lay so many eggs, then you won't have progeny, right?
Yeah.
That's the trade-off.
Yeah. Okay. Thank you. Okay. Any more? Maybe we will open one more questions. And is there anyone who wants to ask questions?
Can I ask a question, please?
Okay, please.
I'm sorry I missed some of your first slides, but I just want to ask about does this mechanism of you explain about the infection of Herpetomonas muscarum into the Drosophila also applies for Leishmania in sunflies? I mean, I'm just curious when this Herpetomonas muscarum doesn't really infect human, then why you perform this experiment to this Herpetomonas instead of doing this directly to the sunfly?
So for two reasons. The first is that sunflies and Leishmania is a very difficult. So sunflies are very difficult to rear in the lab, and Leishmania is very is very difficult to work with. But especially it's the side of the insect which is the most difficult. So for most of these insects, Phlebotomus and Glossinia, there is no genome. So the genome of Phlebotomus is not published yet. So we used a draft version of the genome that we had to annotate ourselves in order to get more reliable gene expression. So bioinformatic resources are in their infancy. Only this year published a paper from NIH where they described a genetic transformation of Phlebotomus. You cannot do genetics, you can only do RNAi, you cannot do all of these experiments with enterocytes and their endocrine cells and so on and so forth you cannot do, basically. So because of the evolutionary relationship, we thought that if we get an understanding of a pair, that we could, it would be it would be easier to get into the mechanistic detail of the interaction. We might be able to go and ask similar questions for the more difficult more difficult to study interaction, but also more important to study interactions of phlebotomus. So we thought that Drosophila would be the gateway to lead us to ask more specific questions about the Phlebotomus leishmania, right? Because if you start with just this, then it's very difficult to know what to look at. So what we want to do now is to try and do either CRISPR-Cas9 or RNAi of STAT, of NF-kappa-B, of DUOX, of these kind of things, and see how these influence blood meal, how these influence leishmania establishment, intestinal stem cells, and so on and so forth. We wouldn't have been able to ask these questions so specifically if we didn't first look at Drosophila.
Okay, thank you very much for your answer.
Oh, okay. Thank you. I think that's the last questions that we can answer. And I would like to say thank you very much to Professor Petros Ligoxygakis that already present us with a really brilliant idea about the use of Drosophila in the examination of host pathogen interaction. And actually, this is what I would like to ask to participants. We previously had around 200 more, and then now we have 160. I would like to ask everyone to turn on the video, and we will capture, capture screen, everyone. Bisa minta tolong di turn on dulu videonya, mau di capture screen untuk bukti bahwa webinar ini telah berhasil dilaksanakan. Maybe after this, Petros, you will know some Indonesian language. Ya, minta tolong Pak Nur Amir dan tim Rifka mungkin. Oke. Sudah Pak?
Sebentar.
Oh ya, ya. Great. Now the one that I already asked would you mind choosing two best questions that you have that someone already asked to you, Petros?
Right. So the was about the the compounds, sort of pharmacological question about the
delivery.
Yes, the delivery.
Okay. So if I'm not mistaken, the first question will be from Mr. Andidian Permana, and the second one is Miss Andi Pudia. Sorry, Miss Andi Pudia. Can you? Sorry, I couldn't see the Andi Pudia Hanum. Is it?
Yes.
Okay. Great, thank you, Petros, Professor Ligoxygakis, for the choose. So for these two persons, please don't leave the Zoom meeting because we will ask some details to give you the present. Okay. Can I ask one more? What do you think about Indonesian audience, Petros? Because now maybe this is the first time for you to have presentation with our Indonesian?
Absolutely. I mean, I was fascinated by the questions. I thought the questions were, extremely high quality. And I enjoyed very much the presentation and the interaction, and thank you for the invitation. I'm very happy to share our work with everyone, and to acknowledge, of course, the fact that these people, so people in my lab had done the work, right? It's very important to acknowledge this, and maybe I should one more time show you the group. There is one person missing from the photo, but it's very important to acknowledge those very, very capable and hardworking people.
That's really, I mean, I always see presentations with acknowledgement, but the way you're saying the acknowledgement is really touching. I mean, I just wondering if you want to, in the future, you want to have Indonesian students in your lab, because in these names, maybe it's not so, some Asians, I think, but not Indonesian.
So we have, Srishti is from India, Nunu is from China, Siv is also from India, so we have a very, Li Hui, who left the lab, was from China. So we have a very international lab. So of course, if students want to, it's a very competitive field, right?
Yeah.
And it's going to be tough. But if you think you have, good students, then absolutely encourage them to apply.
Great, Petros, because we have Indonesian scholarship that can support students to study in any universities in the world. And in fact, one of the person who asked question, which is Miss Dini, maybe you remember the lady that, she actually just got the scholarship and will soon depart to Oxford for her PhD.
So
Yes.
Which lab?
Maybe if she's still here, I hope, Miss Dini?
Yeah.
Yeah, which lab?
I'm joining the clinical medicine, Department of Clinical Medicine, and my supervisor is Professor Persevenborough. I will work with HIV.
Yeah, okay.
Yeah, from Drosophila, she moved. So you're not Drosophila worker anymore, Dini.
Try to diversify my knowledge.
Great. Yeah, I hope the good journey to, maybe you can visit Petros lab soon.
Yeah,
I mean in the future.
Yeah, absolutely. I want to meet maybe Professor Petros.
Yeah, absolutely. Yeah.
Yeah, let's
Cool.
meet first.
Cool. Thank you very much. I think this is the end of our session, I mean event. It is really nice to have you, Petros, and I couldn't say more to show my gratitude that you already, present and something that you did with your student in the lab, and this is really important for us because I hope my students and everyone's students in here is enjoying the presentation, and then they also eager to do the same, like doing something fruitful or beneficial in the future.
Thank you very much. Thank you very much, Farzan, for the invitation. It was, I was very happy to share our work with you. Thank you.
Thank you very much, Petros. I will contact you soon after this. We have something. Okay, everyone, I hope you can, say something or rising, this is the parting moment. So thank you very much, Professor
you.
Petros. I'm really happy to have you here.
Thank you. Bye
Bye
bye.
bye.
Bye bye.
Bye bye. Thank you very much.
Thank you so much.
Bye.
Oke, terima kasih Bapak Ibu atas kehadirannya. Untuk yang 2 orang yang berhasil memperoleh